Assalamu alaikum everyone. Hope you all are doing well. So today I'm gonna discuss something important regarding your biochemistry practical exam. You all might be familiar with your practical exam pattern, that there are two parts of the practical exam. One is OSPE and the other one is long practical. Today we will talk only about the long practical. Actually, the small portion of this long practical. So let's begin. Let's suppose you are in the long practical and you'll be given an unknown solution. You have to identify the compound present in it. You will be given a solution that will be labeled as carbohydrate or protein or lipid or urine. But today we are gonna discuss here the unknown carbohydrate solution only. The other unknown solutions will be discussed later on. So coming towards the topic, you will be given an unknown carbohydrate solution. So how will you proceed further and which tests to perform in sequence wise to reach that unknown carbohydrate to reveal the identity of the solution. So which test you will be performing and why will you perform those tests and where to stop. We will be discussing all these things. So first of all, starting with the scheme of the carbohydrates. So first of all, when you will be given the unknown solution, the first test that you will be performing would be the Molish test. Molish test is a group test for the identification of carbohydrates, which is given positive by all carbohydrates. For example, monosaccharides, disaccharides, polysaccharides, or even the compounds containing the carbohydrates, for example, glycoproteins and glycolipids. And if the Molish test is negative, it means that the carbohydrate is absent, and the other compounds that might be protein or a lipid. So when the test is given positive, which test we will be performing further is the iodine test. Why we perform iodine test? We perform iodine test because iodine test is a group test for identification of polysaccharides. It is given positive by polysaccharides and is negative for monosaccharides and disaccharides. And in case of polysaccharides, these polysaccharides can further be identified by their colors. For example, starch gives blue color with iodine, dextrin gives purple color, and glycogen that gives reddish color with iodine. So if it's starch, so let's suppose the unknown solution was a starch and you have seen that the result of the iodine test is blue, so you will easily identify it as starch, so you will not proceed further. Okay, so if the iodine test that is negative, then you will proceed further. That might be a monosaccharide or a disaccharide. So the next test we will perform the Benedict's test. Benedict's test, why we perform Benedict's test? Benedict's test, this is a test for the reducing sugars. So it is given positive by reducing sugar. That might be the monosaccharide or the disaccharide. And it is given negative by non-reducing sugar. The example of the reducing sugar, the monosaccharides are glucose and fructose, and the disaccharides are the maltose and the lactose. On the other hand, if the Benedict's test is negative, the example is sucrose. We will further perform the hydrolysis of the sucrose that will hydrolyze it into glucose and fructose, and then we can perform the Selivanov test further. And Selivanov test will be given positive because it also contains fructose. So on the other hand, there is a Barfo test. So why we perform Barfo? Barfo test is used to differentiate between monosaccharides and disaccharides, and the test is given positive by monosaccharides within 5 to 7 minutes, which is known as early positive, while disaccharides give it positive within 7 to 15 minutes, which is known as late positive. The example of monosaccharides are glucose, galactose, and fructose, and the late positive disaccharides are maltose and lactose. The next test performed is the tolerance test. Tolerance test, when it is performed on the monosaccharides, so it is given positive by galactose and negative by glucose and fructose, and when it is performed on the disaccharides, it is given positive by lactose and negative by maltose. So the next test which is performed when the toluene is negative, that is the Selivanov test. Selivanov test, why we perform Selivanov? Selivanov test is performed when we want to differentiate between the aldo sugar and the keto sugar, and test is specifically for the keto sugar and is given positive by keto sugar. So Selivanov test is given positive by keto sugar, and the example includes fructose, and when it is given negative by glucose. So the further test that we can perform on the both fructose, glucose, that is the Osazone test. Osazone test is basically used because we can further confirm it by the shape of the crystals of the Osazone test, that either it is fructose, galactose, lactose, or maltose, because galactose gives star-shaped needle crystals, and the fructose that gives the broom-shaped needle crystals. The Osazone test for the lactose that gives the fluffy ball-shaped crystals, and the maltose gives petal-shaped crystals. So if you have the glucose or the fructose, or the last test is Selivanov, you don't need to perform the Osazone test further most of the time, but if required you can perform it to further confirm them by the presence of the shape of the crystals. Hope you like the video. Keep watching my channel. Thank you.